This is part two of the lecture on genetics. In this part, we will talk about um, abnormalities that occur primarily on the sex chromosomes as opposed to the autosomes that we had talked about before. The sex chromosomes, again, are one pair of chromosomes that determine the sex of the individual. The X chromosome and the Y chromosome. All ova have an X chromosome. Spermatozoa typically either have an X or a Y chromosome, and the determination of sex is determined based on the spermatozoa that fertilizes the ovum. If the X-bearing spermatozoa fertilizes the ovum, the individual will be male. If the Y-bearing spermatozoa fertilizes the ovum, then the individual will be female. This is a diagram showing how this works. The ova can only have an X chromosome because woman has two X chromosomes, no Y chromosomes to donate to the ova. And the sperm can be either an X-bearing sperm or a Y-bearing sperm. If an X-bearing sperm fertilizes the ovum, then the individual will have two X chromosomes, will develop as female. If a Y-bearing sperm fertilizes the ovum, then the individual will have an XY chromosomal makeup and will be phenotypically male. So the father is the individual that determines the sex of the offspring, not the mother. The X chromosomes and Y chromosomes look different also karyotypically. The X chromosomes are very similar to the other chromosomes. The Y chromosome is much shorter, carrying much less genetic information. But some of that information that's carried on the Y chromosome is what's going to determine the male sex. There is one gene on the Y chromosome that is not present on the X chromosome and is called the SRY gene. The SRY gene, which is, here's an X chromosome, there's a Y chromosome, is on the Y chromosome. It is not present on the X chromosome. And if fertilization of a sperm with an Y chromosome fertilizes the ovum, that SRY gene, and what do genes do? They code for proteins. This codes for a protein or several proteins called transcription factors that basically produce male structures and reduce female structures. If the SRY gene is not present, then the individual will develop as female. If the SRY gene is present, then the individual will develop as a male. Again, the X chromosome looks similar to the other chromosomes. It contains about 1,500 genes, and again, you have a homologous pair of them in a female. In a male, you do not. You have an X chromosome and then a much shorter Y chromosome. The Y chromosome is much shorter than the X. It only carries about 231 protein encoding genes, one of them being that SRY gene that produces male phenotypic structures. There are inheritance patterns that are based on the X and Y chromosomes. These are called sex-linked inheritance. Because the Y chromosome is much, much smaller than the X, there are many genes that are present on the X chromosome that do not have a corresponding gene on the Y chromosome. Most diseases in sex-linked inheritance are diseases of the X chromosome. There are some on the Y, but they are rare because the Y chromosome is much smaller. And most of these diseases are actually recessive in nature, which means you have to have both of the alleles in the female to have the disease. In the male, you only have to have one of the alleles because of the fact that the Y chromosome does not have a corresponding allele to that. What does that mean? basic rules of sex-linked recessive inheritance. A father who has the disease will pass the allele on to all of his daughters, some of which may not display the disease. They may be carriers if the mother is normal, but he will pass on the disease to none of his sons, because all of his sons are going to get his Y chromosome, which does not have the diseased X abnormality. If a mother has the disease, she therefore must have both of the alleles, and she will pass on that allele to all of her children. If her sons will have the disease, her daughters, if the father is normal, will be carriers. Examples of X-linked recessive disorders: color blindness. Color blindness is basically the most common form of color blindness is a condition that is X-linked recessive. That means it's on the X chromosome, and it is a recessive trait, which means that in a female, if a female does not have color blindness, then her genotype must be, if big C is normal and little c is colorblind, then a normal female is going to be either big C, or big C, little c, because the little c, the diseased allele, is going to be masked by the dominant normal allele. So if the mother is homozygous dominant or heterozygous for these alleles, she will have normal vision. But if she's homozygous recessive, if she has little c, then she will be colorblind. These are the females. In the males, there's two options. Either the male can have the normal allele and the Y chromosome, in which case he will have normal vision, or the male will have the abnormal allele and the Y chromosome, and he will be colorblind. Here's an example here showing this. Here you have a mother who is a carrier. What does that mean? That means she has these alleles on her sex chromosome. She is big C, little c. And the father is normal, which means he is big CY. So here's the mother with big C, little c here. Here's the father with, he's normal with big CY. So neither of the parents are colorblind. The mother is a carrier, but she is not colorblind. The father is normal. If you fill out a Punnett square, like we've done before, then this half of these two boxes represent their daughters, and these two boxes represent their sons. So of their daughters, one of the two boxes, or 50% of their daughters, are going to be normal and have a normal genotype, unlike either of the parents. If the other 50% of their daughters will also not be colorblind, but will have a genotype similar to mother's and can pass that allele on to their children. So all of the daughters, 100% of the daughters in this case, are going to be normal and have no color blindness. But 50% of the daughters will be carriers like the mother that can pass the disease on to the next generation. These two boxes over here represent their sons. Of their sons, one of the two boxes has the normal allele in the Y, which means 50% of their children will have, or their sons will have normal vision, and the other 50% of their sons get the recessive allele from mom, the Y chromosome from dad, and will be colorblind. So of their sons, 50% of their sons will be colorblind, 50% of their sons will be normal. Of all their children, 75% of their children will have normal vision, 25% of their children, or half of their sons, will statistically be colorblind. Other X-linked recessive disorders like Duchenne muscular dystrophy and hemophilia A, which we'll talk about in a few minutes, work the same way. Hemophilia A is a clotting disease caused by a deficiency in one of the clotting factors. Which factor? It's factor 8. Where was factor 8? Well, if we look back on this chart here, here's factor 8 here in the clotting cascade that basically activates factor 10, which then ultimately causes a clot to form. If this is not present, then you can't get the activation of factor 10, and ultimately you end up with no clot formation. And if you get damaged blood vessels, you end up with severe, excessive, in many cases, life-threatening bleeding. Hemophilia A is an X-linked recessive disease, which means that, again, if the mother is a carrier, she can pass that on to half of her sons. Each of her sons, if the father is normal, have a 50% chance of receiving the recessive allele from the mother. Since the son does not have a allele on the Y to mask it, then half of their sons will show the disease. If the mother is a carrier, she will pass on the allele to 50% of her daughters, but each of her daughters will be a carrier. She will not have any daughters that end up with the disease in this situation. One of the factors of X-linked recessive inheritance patterns of diseases is that they tend to show up much, much more commonly in the sons than in the daughters. This is a diagram showing a couple of examples. This first example over here shows a father who's hemophiliac. He has the disease that's shown in red, and his other allele is a Y allele. So he is a hemophiliac. He has a bleeding disorder. He has children with a woman who is normal, and they use black to determine the normal allele. So she has two normal alleles. He has an abnormal allele and a Y chromosome. And this is the outcome of their children. This is an unusual way of looking at it. Basically, what this shows is that he is going to donate this Y chromosome to sons who are only going to get a normal X chromosome from their mother. So all of his sons will be absolutely normal. But he's going to donate his X chromosome, which is where the abnormality is, to all of his daughters. So all of his daughters will not be hemophiliacs, but will be carriers that can then pass on the disease to the next generation. This is a kind of a difficult way of looking at it, so I've made a Punnett square showing the same thing. Over here we have big H being normal, and this should be little h being hemophilia. Let me see if I can fix that now. There we go. Okay, so as we said before, the mother is normal. So her two alleles are big H, big H. The father is a hemophiliac. So he has the alleles little h and y. He has a bleeding disorder. And if we fill out the Punnett square, what we see is we get big H, big H, little h, big H, y, big H, y. These two boxes represent their sons. These two boxes represent their daughters. So in this situation, what you see is that all of their daughters are not going to be hemophiliacs, but they're all going to be carriers that can pass the disease on to the next generation. All of their sons, because their sons are going to get his Y chromosome and the mother's normal X chromosomes, are going to be normal. So the sons will be normal. So on 100% of their children will not be hemophiliacs. 100% of their sons will be genotypically normal. 100% of their daughters are going to be genotypically carriers or heterozygotes for that condition. Another example shown over here. Here's an example of a father who now is no hemophilia, which means he has a black X in his Y, and the mother is a carrier. So the mother is one of these produced. One of these children basically has children with a normal male. So if we take her now as the mother with a normal allele and an abnormal allele for her two alleles for the sex chromosomes, and a father who is normal, who has no disease, and we decide that, okay, he is going to donate, half of his children are going to get his X chromosome and be females, and half of his children are going to get his Y chromosome and be males. So again, if we draw a Punnett square here, what we see is, in this case, the mother is now a heterozygote. She is not hemophiliac, but she carries the disease. The father is normal. He has a big H and then his Y chromosome, which does not have a corresponding allele. Fill out the boxes, and what you see is, this is their daughters, these are their sons. Of their daughters, none of them will be hemophiliacs, but half of their daughters will be normal. The other half will be carriers like the mother. Those are their daughters. These are their sons. Of their sons, one half, or one of the two boxes, 50% of their sons are going to be normal. The other 50% of their sons are going to be hemophiliacs. Probably one of the most famous examples of hemophilia A is in the early 1800s royal family of Queen Victoria and Prince Albert. Queen Victoria, very famous Queen of England for many, many years, a very interesting individual. She was a carrier. She received the allele from her parents, but she did not have the disease. Carriers here are shown in white. Normals are shown in blue for the males, and normal females are shown in green. So here we have Queen Victoria, who's a carrier, and Prince Albert, her husband, who was normal. This is the exact same example we saw here. This could be Queen Victoria. This could be Prince Albert. He was normal. And if you look at that, what you'll see is, of their daughters, none of them will be hemophiliacs, but statistically half of them will be carriers. Of their sons, half of their sons will be normal. The other half of their sons will be statistically hemophiliacs. If we look at the offspring, what they had was they had 1, 2, 3, 4, 5, 6, 7, 8. They had 9 children. Of those 9 children, the first one was Prince Edward, who, being the first male, was in line for the throne. And he turned out to be a normal male, which means that if this is Queen Victoria and this is Prince Albert, this was his genotype. He got lucky. He did not get this genotype, which would have given, excuse me, he got this genotype. If he'd gotten this genotype, he would have gotten the disease. Their next child was a girl, Victoria, named after her mother, the Queen, who was also normal, which means that, again, if we go back to the drawing, she got these alleles. She was genotypically normal. So this was hers, this was her older brother's. So the first 2 children did not show the disease. The third child was a daughter, Alice, and Alice turned out to be a carrier like Queen Victoria, which means Alice, got these alleles like the mother. So Alice got the same alleles that her mother had. What did that mean? Well, that meant that when Alice then had children with her husband down the road, one of her sons, Frederick, ended up a hemophiliac male. They had two other sons after that, both of which were normal, like Edward. But then their next son was Leopold. Leopold was a hemophiliac male, which means that he got the hemophilia allele from his mother, the one that she was carrying, and the Y chromosome from the father. So he was a hemophiliac male, and he had children with a normal female. And if we go back and look here, here's Leopold. Here's a normal mother. And of those, you can see that none of the children will have the disease, but all of the daughters will be carriers. And if we look at Leopold's children, we saw one of his children, and this doesn't list all of them, was Alice, who was a carrier, who then had children with a normal male, and sure enough ended up with another hemophiliac male in their offspring. So she carried it and passed it along to this individual who got the disease. They then had two other daughters who were normal, and then they had their last child was Beatrice, who was also a carrier like mother, who also had children with Henry, and they had two hemophiliac male children, one carrier female, Victoria, who then had two more hemophiliac male and passed that gene on to this generation. So basically what you can see is that on this chart, X-linked inheritance, X-linked recessive inheritance shows up largely in males, and oftentimes it shows up being passed from carrier females, shown as white circles here, that are normal, don't know they have the disease, but they pass the disease on to half of their sons. Another example of the same chart, this to be just more complete. Here's George VI here. Again, this is the line of succession for the British monarchy because this was the firstborn male child, and therefore there is no hemophilia in the British royal family at this point. If we go from George VI to Elizabeth II, which is the Queen now, and then ultimately Queen Elizabeth II with Prince Philip had 4 children, the firstborn male being Prince Charles, who then had children with Diana, Princess of Wales, and his firstborn child was Prince William, who now has several children with the Duchess of Cambridge, and his second born child was Prince Henry, who's now married the former Meghan Markle, and they now have a child. So this is kind of the finishing up of the royal family at this point. But suffice it to say, there's no active hemophilia in the royal family anymore because it has been lost back when their firstborn child here, Edward, was free of the disease. So unless someone marries into it, it will not be present anymore. Finally, we're going to talk about some effects of gender on phenotype. Phenotype is the appearance. There are such things as sex-limited traits. What does that mean? That means traits that you may inherit, but they're only produced phenotypically if you are either a male or a female. What does that mean? For example, the presence of development of a beard. The alleles for beards only grow beards in males. Likewise, the development of breast at puberty is only present in normal situations in females. There are other alleles that are called sex-influenced. That means one allele is dominant in one sex but recessive in the other. An example of that is baldness. In baldness, the allele for baldness is dominant in males, but it's recessive in females. What does that mean? That means that if you are a male, if you get one of the alleles, you will be bald, develop male pattern baldness. But in females, you have to inherit both of the alleles to show that trait. Finally, we've talked so far about abnormalities in the specific genes and different alleles that can cause certain diseases. We're going to finish up by talking about conditions in which there are abnormalities in the actual number of chromosomes. We said that normally all of your cells, besides the sex cells, have a total of 46 chromosomes. But we said we actually think of that as 23 pairs of homologous chromosomes. And actually, we say that's actually, more specifically, 23 pairs of autosomes and 1 pair of sex chromosomes. XX if you're male, XY, XX if you're female, XY if you're male. Under normal conditions, all of the cells of your body have that. That is called the euploid number of chromosomes. 46 chromosomes, 23 pairs. But there are conditions in which abnormalities during meiosis cause an abnormal number of chromosomes. The most dramatic is called polyploidy. Polyploidy means you actually have an entire extra set of chromosomes. Instead of 46, you have 92. It results from the formation of a diploid rather than a haploid germ cell, either a sperm or an ovum. These individuals do not usually survive to birth, or if they do, they die very shortly after birth. They have severe problems with their development. Most of them actually do not survive to birth and are miscarried early in pregnancy. Aneuploidy, however, is a condition in which instead of having a complete increase or decrease in the entire number of chromosomes, there is an increase or decrease in one chromosome. Remember, you have 46, but if an abnormality occurs during meiosis in one of those chromosomes during meiosis, you can end up with a condition of an extra chromosome or a decreased number of chromosomes. If you have an extra chromosome, it's considered trisomy. That means you have a third version of one of the chromosomes. And monosomy is a condition where you have actually one less of the number of chromosomes. You will have 45 chromosomes. Trisomy, you'll have 47 chromosomes. How does that occur? Well, normally in meiosis, basically what happens is meiosis consists of two divisions. The first division, the homologous chromosomes separate. They have each made a copy of themselves. The second meiotic division, the centromere splits, and you end up with gametes that have 1/2 the number of chromosomes that you started off with. Abnormalities can occur either in the first or the second meiotic division. If in the first meiotic division there is an abnormality, then these two homologous chromosomes, which code for the same genes, are normally supposed to go one this way and one that way. But in the first meiotic division, if a condition called non-disjunction occurs, they don't separate, you end up with one produced that has neither of those chromosomes in it, and another one that has both of them. The centromeres then split, and you end up with, ultimately in this case, two of the spermatozoa having an extra chromosome or being trisomic, and two of the spermatozoa produced having one less chromosome after fertilization. In this case, this is a monosomatic condition. So what happens is, in this case, you have one extra chromosome, and this one you have one less. So if non-disjunction occurs during the first meiotic division, all of the individuals produced are going to be abnormal. 50% of them are going to have an extra chromosome, 50% of them are going to have one less chromosome. The other possibility is that the abnormality occurs in the second meiotic division. The first meiotic division goes normally, the homologous chromosomes separate, and then in the second meiotic division, the centromeres are supposed to split, and these copies are supposed to go into individual cells. In this case, it happens normally, and you end up with two cells with the euploid number, which then fertilize the egg, and you have a normal individual. But in this case, they don't separate. You end up with both of them over here, and you end up with a condition after fertilization where you have one extra chromosome or a trisomic condition here, and one less chromosome or a monosomic condition here. So if non-disjunction occurs in the first meiotic division, all of the individuals produced are going to have abnormal chromosome numbers and abnormalities. If the non-disjunction occurs in the second meiotic division, statistically half of the offspring are going to be normal. The other half are going to be either trisomic, a quarter of those, and another quarter will be monosomic or have one less chromosome. Examples. We're going to talk about examples of aneuploidy of the autosomes, and we're going to also talk about some aneuploidy of the sex chromosomes. Probably the most common aneuploid condition that most of you are aware of is trisomy 21 or Down syndrome. Down syndrome means that during non-disjunction, there ended up with a third copy of chromosome pair 21. That's why it's called trisomy 21. So if you do a karyotype on any of the cells of somebody with this disease, you will get normal autosomal chromosome pairs until you get to 21, and you'll have an extra one here. You can see there's 3 of those. Why is there 3 of those? Because somewhere, either in the sperm or the ovum, there was a non-disjunction, and you ended up with an extra chromosome fertilizing the egg, and you end up with a third pair of chromosome 21. Here's chromosome 22, and then here's the sex chromosomes X and Y, the small y indicating this is a male. What happens when you end up with this third set of chromosomes? Well, you end up with the array of different anatomic and structural abnormalities associated with Down syndrome or trisomy 21. And those can vary from something simple like widening of the gap between the first and second toes, the increased fold over the eye or epicanthal folds, the simian crease along the palm. It's an abnormal crease in the palm. Those are all obvious structural abnormalities, but more severely, these individuals can have various degrees of mental retardation, some of them very severe, some of them very functional. They can also have congenital conditions affecting their heart, their major blood vessels, and their gastrointestinal tract. Some of these individuals are highly functional and can live long, productive lives. Others are very, very severely debilitated and require care throughout their entire life. There are other trisomic conditions that are much less common. I will mention a couple of them here. Trisomy 18 is basically a third copy of chromosome pair 18. Trisomy 13 or Patau syndrome is a abnormality or a third copy of the 13th chromosome pair. So if we go back to this, in someone with trisomy 18, you look at chromosome pair 18, which is here, you would have a third one. And chromosome pair 12 is up here. You would have a third one there. So those two are other autosomal trisomic conditions. They're caused by the same non-disjunction occurring during meiosis of the development of either the sperm or the egg. Those are both much, much less common, much, much more severe. Almost none of these children live beyond a year of age if they even survive to birth. Down syndrome, obviously infinitely more prevalent than the others. You can see the incidence of Down syndrome, 1 in 800 to 1 in 826 births is a Down syndrome child, with increased risk at a more advanced maternal age. But 85% of those survive after birth, many surviving many, many years if they're highly functional and have minimal abnormalities structurally. The last conditions we're going to talk about are trisomic or monosomic conditions associated with the sex chromosomes, the sex chromosomes being the X and the Y. The first one we're going to talk about is Turner syndrome. Turner syndrome is a condition of a monosomic state where you have an X chromosome, no corresponding Y or X chromosome. In other words, you only have one sex chromosome and it is an X. Because there's no Y, so there's no SRY gene, these individuals will be phenotypically female, but they will have abnormalities. They will tend to have short stature, underdeveloped breasts, underdeveloped ovaries. They usually do not menstruate. They usually cannot have children, but they have normal mental capabilities, physical capabilities other than the ability to procreate, and they can live long and healthy lives. Remembering that since these are all conditions that are produced at birth and that every cell in your body has this isn't something that can be genetically changed. This is every cell in your body, if you look at someone with Turner syndrome, is going to have this exact same karyotype. Klinefelter syndrome is a trisomic condition in which, when you look at the karyotype, you have two X's and a Y. You have a third sex chromosome, again, caused by the same condition, a non-disjunction. Since there is a Y chromosome with an SRY gene, these individuals are phenotypically male, but they have associated findings because of having that X chromosome. They tend to be tall, they tend to be somewhat mildly mentally retarded in many cases. They have reduced secondary sex characteristics. What does that mean? That means the male sex characteristics don't develop normally at puberty. The testes seem to be small. They generally have infertility issues. They don't produce a lot of sperm. And because of that extra X chromosome, they sometimes can develop gynecomastia, which is a term for breast tissue formation in the male, which is obviously abnormal. Jacob syndrome is another trisomic condition, but this is a condition where if you do the karyotype, you will have one X chromosome and two Y chromosomes. This condition is caused primarily by non-disjunction in the development of the Y chromosome in the male, sperm, because the female does not have a Y chromosome, so this is largely produced by the father. The father with two Y chromosomes in a sperm abnormally fertilizes an ovum that has an X chromosome, and you end up with this condition. These people tend to be tall. They tend to have a lot of severe dermatologic problems, acne. They can have problems with speech and learning. At one point, it was suggested that these people tend to have increased violent or aggressive tendencies. In fact, there have been several TV shows where People have tried to defend people's behavior in court, saying that because they have Jacob syndrome, they are not responsible for their actions. But what they've done is many studies, and they've shown that there's really not an association between this and increasing aggressive or violent behavior. And then poly-X females is also a trisomic condition, but in this case, it's caused by an extra X chromosome. Generally, these poly-X females will have 3 X's, very, very rarely will have 4. This is extremely uncommon. But poly-X females obviously are phenotypically female because they have no Y chromosome. They are generally fertile. They are generally functional, and they usually can have normal children. Poly-X females with 4 X's have severe mental retardation, but again, that's extremely rare. How do we find out about these genetic abnormalities? Well, for many years, if you needed to find out if a child developing had these abnormalities, early on you had to do what's called chorionic villus sampling, where they would actually insert a tube through the cervical os and then take a sample of the chorionic villi in the developing fetus that they could then isolate the fetal cells and get an idea if there is a genetic disease present. Amniocentesis was also done for many years, where a needle would be inserted in the amniotic fluid, which has the fetal cells in it, and those cells could be then analyzed. Nowadays, by about 15 weeks gestation, we can actually draw blood from the mother and actually separate out fetal cells that have somehow circulated from the through the placenta into mommy's blood, and isolate the fetal cells and look for diseases that way, which is the much more common way of looking for those types of diseases today. Again, fetal cell sorting, it says not yet established, but generally by 15 weeks, and some people say by 12 weeks you can do this by looking at maternal blood. Amniocentesis, generally you have to have enough amniotic fluid, again, between 14 and 16 weeks. All of these allow you to do karyotypes. All of these allow for testing against abnormalities, diseases such as Tay-Sachs disease that are genetically inherited, and so forth. Ultrasound is more of a non-specific, looking for abnormalities in growth, looking for relationships between head size and body size, as for the normal development of the fetus in utero. That ends the talk on genetics, and that ends the material for the course. Please contact me if you have any questions. I will be uploading some practice genetics problems, and we will be I will be in touch with you on Blackboard for further information regarding the exams, including the third exams and the final exams.